Bismillahirrahmanirrahim. Yesterday we discussed different types of methods for the transportation of large molecules. One of them was the phagocytosis. This phagocytosis is oxygen dependent killing and it requires NADPH. And this oxygen dependent killing is done by the help of different types of enzymes. It is also known as oxidative burst or respiratory burst. What happens in the case when a certain type of bacteria or virus attacks a cell? What happens is that a biochemical mechanism of action takes place in the following steps, and this causes the killing of the specific type of bacteria. First of all, the oxygen is going to be converted into oxygen free radicals with the help of enzyme NADPH oxidase. Because it is energy dependent, therefore NADPH is going to be converted into NADP, and then the free radical formation causes the formation of hydrogen peroxide with the help of superoxide dismutase, and then this superoxide dismutase combines with chloride with the help of enzyme myeloperoxidase. All these oxygen free radicals, hydrogen peroxide mainly, and hydrogen perchloride kills the bacteria, and they act as reactive oxygen species. If these reactive oxygen species would remain in the cell, they will start peroxidation, membrane peroxidation. Therefore, membrane damage will be started. This is the overall mechanism of action which depends on oxygen and requires a series of enzymes for the killing of bacteria, is known as oxidative burst or the respiratory burst. The mechanism of phagocytosis occurs in the following manner. We have macrophages and neutrophils for this process to occur. They ingest and kills invading pathogens by three main steps. First of all, recognition and adherence. Second is the engulfment, and third is the intracellular killing. What happens in the recognition and adherence? First of all, the phagocyte is present on the cell membrane. A specific type of pathogen comes and engulfed by the neutrophil phagocytic cell, and after the engulfment, this is converted into this phagosome is converted into this engulfment causes the formation of phagosome. As the cell already have lysosomes, then this phagosome is going to be combined with the lysosome, and it is called phagolysosome. This causes the killing and digestion of microbe by the same process of oxidative burst or respiratory bursts. Then intracellular killing occurs by the process of oxidative burst, or which is also known as respiratory burst. Here you can see hydrogen peroxide, nitric oxides are being produced and used, and then this intracellular killing causes the release of indigestible material by exocytosis. So we can summarize the phagocytosis by the recognition, engulfment and intracellular killing of the cell. This is the overall summary which describes the phagocytosis, in which phagosome is going to be converted into phagolysosome and then killing occurred by the respiratory burst. Hope you understand this concept clearly. What happens in the oxidative burst, we use oxygen, which causes the formation of hydrogen peroxides, nitric oxides, and these are required for the killing of the bacteria. Therefore, this is known as oxidative or respiratory burst. The last two concepts for studying the cell membrane or the equilibrium beyond the cell membrane is the Gibbs-Donnan equilibrium. As we already discussed, that active transport of different substances is going to be taking place across the membrane. This is done with different types of pressures. It could be osmosis, it could be osmotic pressure, or it could be Donan's membrane equilibrium. First of all, we will discuss osmosis or osmotic pressure. Osmosis is the process by which, because we mainly talk about water, it passes from a solution of lower solute concentration to a solution of higher solute concentration. Suppose a container, and this container in the mid is separated by a semi-permeable membrane, which is permeable to solvent but not to the solute. Therefore, selective passage of the solution in response to a concentration gradient occurs. This is the simple process of osmosis. And to stop the movement of these different solvent across the semi-permeable membrane is hindered or stopped by a pressure which is known as osmotic pressure. Talking about osmosis, I give you the example of a container having a semi-permeable membrane, and different solute particles are going to be moved across the semi-permeable membrane, and the pressure or the hindrance caused to stop this osmosis process is known as osmotic pressure. Why we are going to study this osmosis and osmotic pressure across the membrane? Here we take the example of a container while it is going to take place in the human body across the cell membrane. We are studying cell membrane, that's why we have to study this osmotic pressure and osmotic. These are the normal processes. But to maintain this osmotic pressure, we have Gibbs-Donnan equilibrium. As we know that unequal distribution of ions occurs across the semi-permeable membrane. यहाँ तक वही वाली बात है, unequal distribution हो रही है across the semi-permeable membrane. But due to the presence of non-diffusible ions, कुछ ions ऐसे होते हैं, कुछ charges, कुछ molecules ऐसे होते हैं जो कि non-diffusible होते हैं, large होते हैं, like proteins. इस केस में क्या होता है, प्रोटीन्स वो मेंटेन करते हैं इस न्यूट्रलिटी को या होम्योस्टेटिक स्टेट को. दैट्स वाई Gibbs-Donnan equilibrium is the extension of to study the osmotic pressure, and this osmotic pressure is maintained with the help of the presence of non-diffusible ions, which are the proteins. And what is the importance of this Gibbs-Donnan membrane equilibrium? As we know that cell contains impermeable ions. Impermeable ions वो होते हैं जो कि semi permeable membrane में से पास ना कर सके. If they were at equilibrium, the ratio of all monovalent cations would be the same. Therefore, there would be an osmotic pressure difference. समझ रहे बात को? Osmotic pressure difference आएगा जो कि cell को swell कर देगा, causing the cell to become swell. Therefore, active transport maintains a homeostatic state in which there is osmotic balance regulating the cell's volume. Here, what is the thing? The impermeable molecule. causes the maintenance of homeostatic state. Therefore, we study the Gibbs. Therefore, we study the Gibbs-Donan membrane, Gibbs-Donan equilibrium, and this Gibbs-Donan equilibrium mainly explains the presence of non-diffusible ions, which are responsible for the maintenance of osmotic pressure across the membrane. Therefore, the cell maintains the homeostatic state of depolarize and repolarization. Therefore, everything is in place. And this Gibbs-Donan membrane equilibrium is very important to study the maintenance of osmotic pressure across the semi-permeable membrane and to attain the neutrality and homeostatic state of different diffusible ions across the membrane.